The purpose of this video lesson is to explain to you something that is called epistasis. Epistasis is a kind of an abnormality that we see in genetics whenever things don't fall into a nice dominant or recessive. So for a lot of times, genetics don't follow the rules that we've talked about, whether there's one dominant and one recessive trait. Sometimes genes can interact. And so this video is about epistasis, in which one gene is going to mask or suppress a second gene. And this is where we actually get different colored squash. So squash that you buy in the store are usually yellow or green, but there is actually a form that are white squash. And many of you are familiar with Labrador Retrievers. So many of you think of yellow labs. There are also chocolate labs, and there are black labs. And so we're going to talk about how that works and the genetics of how that works. So we're going to start off with squash. Sorry if you're a puppy fan. We'll start off with the squash and we'll move to the puppies. So here's the way we define the letters. So there are two genes that we're going to think about, just like we've been working these 16-box problems in this last section. So big A is going to be the dominant. That's a yellow squash gene, so it makes a yellow pigment in the skin. Little a for green squash. So the green is recessive and the yellow is dominant. There is a second gene, the B gene, where big B is going to mask the color gene. And when we say mask, it hides or it turns off that color gene. The little b, there will be no masking, so it's recessive. Now we call this epistasis dominant because this is the epistasis gene. The B is the epistasis gene. Okay. If it's dominant, it does the masking. So the dominant masking of the gene. So in this case, we're going to take two white squash, and they're white because they both have a big B. Now, as it turns out, they're heterozygous for the masking gene. Okay, and these are, so this is heterozygous times heterozygous, and they're both, in fact, if they weren't epistasis, if the epistasis wasn't turning off the color gene, they do have a big A, both of the parents. So they would have actually been yellow. So these are sort of yellow squash that are that are being crossed, but they're white because the epistasis gene, the big B, has turned off the yellow pigment, so it does not produce pigment at all. So then we do the cross, and so just like before, when you're doing big A, big B, big A, little b, so the rule applies just like we were doing for when you're setting up the Punnett square. So we foil the one parent, we foil the other parent, and then we bring the genes together. So if we're interpreting this, remember the epistasis was big B, so anything that has big B, so all these big B's that I'm clicking on, they're white. It doesn't matter what the coloration was, the big A or a little a for the color. Those squash turn out, if we plant those seeds, they will turn out to produce white squash. And never does a plant produce all colors or two different colors. So the plant will either produce all white, all yellow, or all green. But the seeds that are produced inside those squash could grow to be different things. Now, if we look at this squash here, so it's yellow. Why is it yellow? Because it has a big A, so that's the yellow, but it has two little b's, and so the masking gets turned off, so to speak. So there's no masking, so that squash can be yellow, and this one can be yellow, and this one will be yellow because they all have the big A. This plant down here, why is it green? Well, it's green because two things. The epistasis is not masking, it's little b, and secondly, it has little a, which is for green squash. So there's a 1 out of 16 chance. So the ratio here, though, is 12 white to 3 yellow to 1 green. So that becomes the epistasis ratio, 12 to 3 to 1. So the problem works out the same. You just have to make sure you're careful with the letters and know if it's epistasis dominant, and if it's dominant, that does the masking. So in this case, it's dominant, so the big B masks the color. Okay? So let's look at Labrador Retrievers. Okay, so Labrador Retrievers come in a variety of colors. So as I mentioned before, we can have the black. So here, this is a, let me grab a little better color. So here's the black Labrador Retriever that has a black nose. There's a chocolate Labrador Retriever that's going to have a chocolate nose and kind of chocolate lips and actually chocolate paws as well. But then there can be yellow Labrador Retrievers. Now I know this one looks like a darker yellow and this looks like a lighter yellow. I don't think that's necessarily the case. Maybe the ones, so maybe occasionally they can be a little bit darker because of the epistasis, but we're just thinking of it as being yellow. Now, as it turns out, those yellow labs can either have, and this yellow lab is supposed to have a black nose, and this one is supposed to have the yellow nose, or excuse me, not the yellow nose, the chocolate nose. So the black nose and the chocolate nose. Because as it turns out in the epistasis that we talk about this time, doesn't turn off the coloration for the paws and for the nose. So how does this actually work? Well, in this case, we're going to say there's this big E, and I know it's really small to see here, but this is an epistasis recessive. Now, as it turns out, the first gene, and so here's the gene that we have here, little e, and here's little e. Because this is epistasis recessive, those little e's turn off the second gene. So this individual here was little e, little b. This is little e, big B. And we don't really care what that next color is. But in color, big B is black. So our black is the big B, and little b is the brown or the chocolate. So this genotype here, little e, big B, would have been black coat if it wouldn't be for the little e. And that's why it has the black nose. And this one is little b, so it was going to be chocolate, but the only thing we see chocolate is the nose and the paws. Whereas the dogs on the right here, they have a big E. Now remember, epistasis was what masks. Since this is epistasis recessive, the big E does not mask. So the chocolate lab with a chocolate nose, because it's big E, the epistasis gene is not turned on this time. And the big B here would have been chocolate. Or excuse me, it would have been the black lab. Okay, so again, making sure that we just know if it's recessive or dominant, and they'll tell us in any of those problems. So recessive means that it needs to be 2 recessive genes to turn off the coloration. Whereas in our dominant, 1 dominant allele would turn off and would mask the coloration. So just make sure you'll have these to use in these practice problems that you'll work. So let's look at the next problem. So here we've taken, here's our definition. So big A, the masking is off. Big A, the masking is on. Now in this case, this example that I found, the first gene here is the masking gene. The second one is the color, and it didn't use E's this time. And it doesn't matter. It's whatever is being defined. So just make sure you read that carefully. So big A here is the masking is off. Little a, the masking is on. So and big B was the black pigment and little b for the chocolate pigment. So what does this parent look like? Well, big A, the masking is off. Big B, little b, it's a black lab. So this is a black lab. And as it turns out, it's actually not a purebred because it does actually have the chocolate inside of it because the little b is hidden. This is also a black lab because the big A, the masking is off. So this will be a black lab. Okay. So we cross them. So we do the Punnett square works just the same. So big A, big B, you do the foiling. So whomever did this, the foiling is done just the same, top and bottom. Then here's what things are going to look like. So now let's look at the masking is off. If it has a big A, then the masking is off. So all these ones that are black or chocolate, they have at least one big A. So that means that the coloration comes through. So all the blacks have one of these big B's. And they will have a black coat and a black nose. The chocolate, this is chocolate, this is chocolate, and this is chocolate because they have the little B's, the chocolate pigment. Now these 4 down here in the bottom right-hand corner, why do I have a yellow box around them? Those are going to be yellow labs because remember little a, the masking was on, and they have little a, so the masking is on. The color gene is being masked. Now, all of these pups right here, what will they all have in common? Since they're big B, big B, they would have been black, but they're yellow labs now, but their noses will all be black. Now, I don't think that I have a brown color, unfortunately, but this guy right here, okay, this pup, would be the one yellow lab that would have a chocolate pigmented nose and paws, because the little B's would have said it would have been chocolate, but because it has little A's, the masking gene is on. So those are epistasis recessive. So hopefully that does make sense. And if you need to go back through and watch that again, and there are actually lots of videos out there if you'd want to, YouTube it and see some other things. Hopefully my explanation is adequate. Now the remainder of this packet, then you have a couple of problems. You have a squash problem. So when you work the squash problem, if you need to refer back to the squash stuff, do that so you can see the example here. So I've given you some original squash that I've crossed. Okay, over here are the genes I've defined. So this is asking you, what do the original squash look like? What's their phenotype? So tell me, are they green? Are they yellow? Are they white? So look at each of these, and you're going to say like white times white or whatever they might be. Then you're going to do the Punnett square on the next problem. So foil it. So foil the parents. Then do the Punnett square. And then figure out, please, the ratio of white to yellow to green. Okay, and I'll have posted a key on the board, or you can check with somebody else who's been doing this problem. Then, okay, there is a lab problem. So epistasis recessive, over here are our genes. I've defined them for us. And here are a couple of pups that we're going to cross. Okay. Now, as it turns out, if you'd ever cross a yellow lab with a yellow lab, because they'd be both little a times little a, you'd always get yellow pups. And that's often why we get a lot more yellow labs than all the other ones, because if you do ever cross any yellow labs with each other, they will all be yellow pups, but they could have different color noses. But in this case, we do not have two yellow labs, but we might get some yellow pups. So cross these two. Again, what do the parents look like? Then do the Punnett square. So I've provided a Punnett square for you. Just foil it like normal. Then figure out the ratio of black labs to chocolate labs to yellow labs. Okay, and again, I'll post the answers to that or check with a neighbor. So hopefully that helps understand in case you see one of these problems. And if you ever wondered where yellow and chocolate and black labs came from, you now have a better idea. Thanks for watching.